Hi and welcome to SBP 4504. In this video, we will be explaining on the topic of genetic basis of drug metabolism that have been prepared by the HAVO. These two objectives will be the main focus in this topic. Before we go into details, what is meant by pharmacogenetic? Pharmacogenetic is a study of the role of genetic in drug response. And what is meant by genetic polymorphism? Genetic polymorphism can be defined as when a place in the DNA sequence where there is variation and the less common variant is present in at least 1% of the people. Here is our first example, which is G6PD. G6PD, or known as glucose 6-phosphate dehydrogenase, is used to maintain oxidation reduction in the cell. The absence of it will lead to deficiency and allow oxidative drug to oxidize and cause hemolysis. G6PD deficiency is a sex-linked recessive trait, and it is caused by one-point mutation where nucleotide conversion from cytosine to guanine occurred. The low activity of it resulting in reduced glutathione concentration in erythrocyte and subsequently caused hemolytic anemia following the ingestion of certain drug. G6PD deficiency is divided into two types of mutation, and it can be found commonly in African and Mediterranean lineage. Here is the list of drugs that cause hemolytic anemia for G6PD deficiency. In order to treat the cause, cessations of drug administrations is needed, and for severe cases, blood transfusion and corticosteroid administrations is required. Okay, let's move on to the next example, which is an acetyltransferase. N-acetyltransferase is a phase 2 conjugating liver enzyme which catalyzes the N-acetylations and O-acetylations of amine carcinogens and heterocyclic amine. Na2 gene is responsible for fast acetylator phenotype. When 2 or 3 point mutations occur, it results to slow acetylator phenotype. When someone is having a slow acetylator phenotype and ingesting these drugs, it will increase the risk of getting peripheral nerve damage, idiopathic lupus erythematosus, and increase the risk of getting cancer. People with slow acetylator phenotype are most commonly found among Egyptian and Jewish, while East Asian and people at the north part are the least common.